And we'll get started. We're really excited to start our invasive species awareness series with our keynote presenter, Dr. Michael J. McTavish. And we have a couple watershed staff with us today as well. My name is Nicole. I'm the freshwater health coordinator. And we also have Monica, who's the communications and fundraising manager. And if you're having any technical issues throughout the presentation, you can feel free to send Monica a private message through Zoom and she'll help you with those. A bit about Watersheds Canada. We're a nonprofit and charitable organization based in Perth, Ontario. We deliver programs across the country in partnership with landowners, community groups, and students who are looking to take action to protect their local freshwater. So on the screen, you can see a couple of our different programs. On the top left, we have our Natural Edge program. We work with community groups and shoreline owners to naturalize their property using native plants. On the bottom left, we have our Love Your Lake program, which is delivered in partnership with the Canadian Wildlife Federation. It's a shoreline evaluation program where we fill out an assessment for each property on a lake, and each property owner gets custom recommendations that are voluntary for their property to help protect their lake. And then on the top right, we have our fish habitat restoration program. So in that photo, you can see one of our winter spawning bed enhancement projects. So we put washed riverstone on top of the ice, and then in the springtime, that drops into place. And finally, on the bottom, we have our nature discovery programming. So this is an outreach program for children and youth, and we help provide people with tools to safely connect them with nature. And we're all here today for our freshwater stewardship community, which is an online community that's connecting people from across the country. So this is our third year of the community, which launched in 2021. And all of the webinars and the handouts have been archived on our website. So you can find 25 webinars and 19 handouts. So I'd encourage you to check those out today. If you haven't seen some of them, we have webinars from the private sector, other nonprofits, academia, and a wide range of topics about freshwater health. And we'd also like to thank the Peterborough KM Hunter Charitable Foundation and the SM Blair Family Foundation for their funding support for the community this year. And I'll just take a moment to let you know about a couple of upcoming webinars later this week. So tomorrow at 11, we have a webinar about water soldier. And on Thursday at 11, we have a webinar about grass carp. So more topics in our invasive species awareness series, and that's exciting. So you can register for those at watersheds.ca/freshwater-stewardship. And part of our invasive species awareness series, we've also created some resources and activities that we're releasing on our social media accounts each day. So you can check those out there. And on Friday, we're going to be releasing our invasive species management toolkit. So we're looking forward to that. I would keep an eye out on Facebook, Instagram, Twitter, and LinkedIn for those. And Monica is going to add our socials into the chat so you know where to find them. And then finally, the presentation that we're here for today, Dr. Michael J. McTavish is a postdoctoral research fellow working with the Smith Forest Health Lab at the University of Toronto. His current research includes biological control of non-native invasive plants, including introduced Phragmites and garlic mustard, and the ecology of non-native earthworms. So with that, I will pass it over to you, Michael.
Great, thanks very much, Nicole. I'm going to pull up my slides here for a second. Let's make sure that's all going all right. We're good, I think. Okay.
Yes, you're all good.
Perfect, thanks very much. So good morning, everyone. Glad you're able to join us here, and a big thank you to Watersheds Canada for hosting this webinar today. So hello to folks joining right now and anybody watching this recorded as well. I know just from seeing some of the names coming in as well, I'm pretty sure I've crossed paths with a number of folks over the years as well. As Nicole mentioned, we've got our hand in a few different pots, including Phragmites and garlic mustard biocontrol and earthworms and a few other topics. So we've potentially run into each other at that time before. And today I'm kind of excited because we don't get to actually talk about the garlic mustard aspect of the project too often. So it's nice to have an opportunity to do that. And so what I'd like to focus on, because there is such a plethora of information out there and resources available for garlic mustard management, we're going to cover some of that, some of the basics, just sort of rehash some of those details. But I also want to focus on some new insights into sort of the ecology and management of this weed that we've been dealing with for a good amount of time here. And I should say that this work represents primarily work done with colleagues Sandy Smith at University of Toronto and Rob Boucher at Ag Canada. So, like I said, this has sort of got three main chunks to it, and the first of these is going to be focusing on some of these basics, back to basics of garlic mustard, looking at these big questions of processes of invasion, impacts, and then a relatively quick overview of conventional control methods. I'm going to be relying pretty heavily on a few resources throughout. If you're looking for more detailed background about garlic mustard, and especially on the management side of things, sort of best practices that are currently recommended, I would strongly guide you towards these and other excellent resources. I'm based in Toronto and Ontario, so of course we sort of slant in that direction a little bit, but a lot of these are going to be transferable lessons as well to an extent. So in particular, I'll be relying on and drawing information and some images as well from the Invasive Species Center Meet the Species page for garlic mustard, the Best Management Practice Guide for Ontario that was put out by the ISC and jointly with the Ontario Invasive Plant Council. And also NASMA has some really nice succinct weed fact sheets and biocontrol sheets as well for garlic mustard and those biocontrol agents. So again, if you're looking for more, there's plenty out there, and you could do worse than to start with what we have here. So of course, what we're talking about today, our weed in question is garlic mustard, or Alliaria petiolata. This is a non-native invasive herbaceous species, very common in forested habitats, although you'll find it elsewhere as well. It's fairly flexible in that regard. We're going to get into the biology and life history a little bit further, but it is a biennial, so you can see here images of both that first year form of these basal rosettes clustered down near the ground, and also the taller second year bolted plant that will eventually put out seed and reproduce. It goes by a number of different common names, including hedge garlic, saucelone, jack by the hedge, and poor man's mustard. And you'll notice sort of an emerging culinary theme from some of these names here. And that really speaks to the fact that our best evidence suggests that this is a plant that was brought over intentionally, for the most part, from Europe sometime in the mid-late 19th century, so 1860s approximately, as a food plant, and also as a plant with some potential medicinal properties as well. In Canada, it's currently found in a number of provinces, so British Columbia, Alberta, Ontario, Quebec, New Brunswick, Nova Scotia, and PEI, and throughout a good number of states in the US as well, with current estimates putting it at over 30. And you can see this fairly familiar plants database image here showing just a province and state level image of that distribution. I will say this can be a little bit misleading to some extent when we just look at this high level overview. So almost a better representation, this is drawn from EDMAP's reporting system, and it's providing information on a sort of county level occurrence. So in green, we're seeing where we have reports of the species. Now, of course, lack of reports does not mean that that they aren't present here, but this still gives us a good general idea of where it is most common. And so what we can see is, in contrast to that province and state level, even though, yes, it is quite widely distributed at a large scale, you can find it in quite a few different places. There are definitely some patterns of stronger concentrations in particular areas. So really centered on the Great Lakes there, we have the highest incidence of reports coming from the Northeast and Midwest, and some clusters in the Pacific Northwest as well. So yes, very broad distribution, sort of at a North American scale, but with some particular hotspots focused in particular around that sort of Great Lakes region as well. So as we said before, garlic mustard is a biennial, so it has a 2-year life cycle, starting out as a first-year basal rosette, and then turning into the second-year bolting plant. I've pulled and condensed this sort of life history image from Jeannie Katovich's led NASMA weed biocontrol weed sheet that I mentioned, and it's sort of really useful for depicting this slightly complex life history. So you can see there on the top we have the progress of in that first year, and then on the bottom in the second year laid out across the months. So basically in sort of mid-late winter, early spring, you can get seedlings germinating from seeds in the soil. And these develop in the early spring into these rosettes. This is a very early developing plant overall, one of the first things that you're going to see that's greening up. And it will persist as that first year rosette form and overwinter, sometimes green, sometimes dying back at the surface under the snow into that second year. And then in the spring again, it's going to bolt into that second year plant, flower, and then mature, produce, and then release very large quantities of seeds, which will then lie in the seed bank for a period of time and feed back, of course, to potential seedlings for following years. I've pulled from that same NASMA weed sheet some photos of different parts of the plant and different life stages here that really capture it quite nicely. So if we sort of look up in the top left there, we can see the very distinctive root structure that we get if we were to sort of grab at that root crown and pull it up. They're usually very easily uprooted, relatively speaking. Continuing along the top to the right, we have a sort of small cluster of some of those first year basal rosettes. Then we have what the leaves look like on the second year bolted plants further up the stalk. So important to recognize from first year to second year that leaf morphology does change. So what you're looking for is a little bit different. And then on the bottom, we see some of those flowers that would appear on a bolted plant. The seed pods or calyx as they begin to mature in this sort of lighter green. And then finally, if we were to crack one of those open, you see some of the seeds lying inside, still sort of partway through development. Those seeds are important because it is the primary, well, it is the exclusive means by which garlic mustard does reproduce. So these figures are very much regionally dependent, but plants produce up to about 22 seeds per each of these pods or calyx. And if a plant's doing really well, it can develop up to in the neighborhood of around 150 pods per plant. So this obviously adds up very quickly to a very substantial amount of seed produced per plant. And if we look at this at more of a population level, sort of a seed density per unit area, we can get a very high seed rain. Again, there are all sorts of different averages out there. One fairly representative average is that you're getting about 15,000 seeds for every meter by meter of forest floor. So a very substantial quantity with values that have been recorded up to and in excess of over 100,000 seeds per square meter. And we'll talk briefly about part of why garlic mustard is so successful and impactful as a weed, and this is certainly a big part of it. Those seeds will mature and begin dropping in July and August, as we saw on that figure on the other slide there, and these can germinate the following year or may remain viable in the seed bank for a period of maybe about 5 to 7 years, depending on local conditions. The primary impacts of garlic mustard that we're familiar with and that we're concerned about relate primarily to overall reduced biodiversity, certainly at that forest floor level in terms of that herbaceous community. And most of this comes from the fact that it is just such a good competitor. Because it is able to outcompete and grow so quickly, it essentially just dominates that forest floor. And so when we have a heavily garlic mustard infested area, we get fairly familiar sites like we have in the top left of a forest floor that is just completely covered by garlic mustard exclusively. There are, again, a number of factors that basically make it a good weed and a good invader. The fact that they're growing very early, generally earlier than anybody else, and they're able to claim that space and claim those resources. There's that that extremely high seed output, which means just that you're getting a huge amount of population growth from that. There's that persistent seed bank. So the fact that they can stay viable in the soil for half a decade, up to a decade even, means that even if they're starting to be outcompeted, there's still new propagules being introduced into that system. And this poses some challenges for management as well that we'll get into also. In its box of tricks, garlic mustard is also known to be allelopathic. So essentially it's altering soil chemistry in the habitats that it's living in that make those conditions less suitable for the growth of other plants. In particular, We have evidence showing that this can inhibit mycorrhizal growth, so fungal associations that plants use with their roots to help in the uptake of water and nutrients. Garlic mustard is not dependent on these associations, so by inhibiting these, it's giving itself yet again another one of these little competitive bumps over other species that do rely on them for nutrient acquisition. There are, of course, many different factors that come together alongside garlic mustard's innate competitive abilities that sort of create the situations in which it does really well or maybe doesn't do as well. We know it benefits quite a bit from disturbance. It doesn't require it, so you can find garlic mustard in fairly intact, say, forest interior environments, but where it is most pronounced would be areas where there has been some kind of disturbance. So a very common example that we see quite a bit would be the edges of habitat. So trails running through forests in particular are quite common. Part of that comes from its ability to use higher light conditions. So it's fairly shade tolerant to a degree, but when it has access to light, such as that outside environment or where we've had some sort of canopy loss, it's really able to take advantage of that and flourish often. I work with earthworms, so of course I can't help but bring up the potential earthworm connection as well. A number of researchers have certainly observed the fact that very often when we see garlic mustard in a habitat, we also see non-native earthworms kind of co-occurring with them. And there's some interesting speculation about potential mutualistic associations between them, where garlic mustard helps the earthworms and the earthworms help the garlic mustard. One of the potential ways in which earthworms are good for garlic mustard is by accelerating the decomposition and removal of that forest litter layer, so sort of decaying organic matter, leaf litter that builds up normally on the forest floor and takes a long time to break down in our forests. Earthworms can make that disappear much more rapidly. Amongst other effects, this exposes that forest soil floor, which then leaves developing seedlings that are used to having that protection vulnerable to desiccation or to browsing by herbivores, including deer in particular. So there's a lot of compelling evidence about potential synergies between non-native earthworms, deer, and garlic mustard, all having sort of a three-hit punch against native forest plant biodiversity. So as a result of these various interactions and its innate competitive ability, we can see garlic mustard in many situations becoming this very dominant understory plant in as few as a few sort of years or up to a decade of arrival in an area. In terms of other impacts, there are also sort of more anecdotal reports of garlic mustard sort of flavoring the milk from dairy cows that have been grazing on it. I haven't seen a ton of sort of more scientific research looking at this, but it's certainly an anecdotal report that you'll get as well. So there are lots of reasons that we will want to try and control garlic mustard then because of all these negative effects. I want to sort of flag here before we get into that there's some very recent and interesting research coming out from Bernd Blossy and colleagues looking at potential declines actually in garlic mustard population growth rates over time. So essentially in their studies of garlic mustard populations that have been established for a very long time versus those that are quite young, what they tend to see as a general pattern is that these older populations have lower growth rates, which even tend towards either stability or maybe even population decline. And there's some interesting ecological reasons as to why this might be happening, including potential plant soil feedbacks that emerge that as these plants are living in this habitat for a certain amount of time, over time, their successfulness there can potentially decrease. And that's good news from a management side of things, but I would say it doesn't let us fully off the hook of this. It's still very important, even if this is happening, to manage garlic mustard where possible and where appropriate to protect sensitive ecosystems, because this is an effect that by its very nature takes time to develop, if it does develop at all. And there can be sort of unacceptable losses during that period of time as well that we may want to offset. So there are, of course, various control methods that you may be familiar with as well. Broadly, these fall into these typical categories. We have physical and cultural methods, such as hand pulling or cutting, clipping, burning of an area. We have chemical strategies that include herbicides, typically fairly broad spectrum herbicides. and also biological control, which is an emerging strategy that we're involved with that we'll be talking about as the last section of this talk here today. So I don't want to be too down about this. Management certainly can be have a lot of benefits and can be very useful in certain situations, these kind of conventional management approaches that we're talking about. It can be really useful for outreach and for building awareness and community engagement with environmental issues. So garlic mustard pulls, even if they don't achieve anything beyond that, just making people aware of that and take some sort of stewardship in an area is in and of itself a really valid outcome. The problem is that oftentimes we see a lot of these methods not really being very effective for control. They usually take a lot of labor, they're a lot of work, they only really work at small scales, and oftentimes, and especially if that pressure is not maintained, their effects can be very short-term, so sort of not long-lasting. Of the methods that we have, I've pulled this from that ISC OIPC Best Management's Guide, and it does a good job of summarizing the control methods that we have. They've arranged them on this table in terms of the size of the infested area, so from just a few plants up to a very large multi-hectare area, to the density of plants in those spaces. And all those methods that we talked about briefly there are arranged here. And what you can sort of see as a general pattern is that basically we get a shift in methods when we move from very small, low density infestations, and that's when we're kind of looking at greater viability for things like pulling and mowing and cutting in particular, more of these physical methods. And then as an area that we're trying to address becomes larger or has a higher density of weeds, that's when we need to start looking at more of our chemical, biological control, controlled burns, sort of more higher severity management tactics. Now, garlic mustard is really tricky to manage at the end of the day, and there are a lot of things that drive that. I think an underappreciated aspect of it is this biennial life cycle. I know from some of the work that we're doing with garlic mustard, trying to track populations over time, because they switch between that first year and second year form, and some populations they will kind of alternate like that. So one year it's a lot of first years, and then the second it'll be second years. Some are more mixed. And so you have this whole sort of range of situations. It can actually be really difficult without a lot of data over many years to get a good comprehensive picture of what that population is actually doing. So you could get a population just suddenly seems to explode or suddenly seems to disappear, but that might not give you a good idea of what it's actually doing over, say, a longer 5 to 10 year period. So monitoring is really challenging. We mentioned that persistent seed bank. So when you have seeds that can stick around for 5 to 7 years, that means that you need to do at least 5 to 7 years of management minimum in order to really sort of deplete all that material that can keep coming out of that seed bank in the interim. So even if the plants that you remove don't have any chance to reproduce, those seed bank seeds are still going to come back and need to be managed. Otherwise, you just go back to square one. Even when garlic mustard is removed, there's also the potential for what we would call legacy soil impacts. So even when the plant has been removed, its impact on the soil community, mycorrhizal communities can still be altered for a period of time that can be quite lengthy as well. And that can make restoration of the desirable vegetation community that we want difficult, even though we have removed the weed itself. So I've pulled up a few additional considerations on the management side of things, again, leaning pretty heavily on that BMP guide as well, that I would suggest everyone check out if you're interested. And the first is really to prioritize where control is and to be realistic about objectives and chances of achieving them. So from that guide, they have a few tables here. The first looks at site prioritization. So basically thinking about situations, when do you actually need to sort of control this? So prioritizing, for example, sites where garlic mustard is not there yet. So prevention is easier than trying to deal with it once it's there, or is just at the very early stages of invasion. This raises important questions that should be happening, discussions about why an area is being managed. Are there rare species or communities that you're particularly trying to protect? Does that habitat have certain land values, productive land values, cultural values that you want to prioritize. And then really taking a moment to evaluate the cost and effort that's going to be required for this. Because of that multi-year sort of requirement, this doesn't happen immediately, and it's not cheap or easy. So in addition to prioritizing sites overall, you can also think about prioritization within a control area. And a lot of these are sort of some of the same ideas, just at a different scale. So really thinking about this less about managing a really dense kind of invaded area. It's really about managing areas that aren't invaded yet. So trying to preserve those as uninvaded, keep them free of invaders, deal with those smaller satellite populations that'll break off from that dense core and potentially facilitate that spread. They're smaller and easier to manage, and you sort of kind of contain the problem as much as possible. And then over time, if things are successful, you can kind of take that uninvaded area and essentially push that inwards and try and reduce the size of that invasion overall. We sort of mentioned this already, but the second big consideration would be the importance of sustaining management for 10 plus years. If you go in and you pull a bunch of garlic mustard once or twice, again, that can be good for outreach, but really you're not achieving anything else. In some cases, that disturbance can actually facilitate even more regrowth from that seed bank and almost make the situation worse. So if you're really trying to do this, recognize and sort of commit to this kind of 10-year kind of time frame to really do it properly. Going hand in hand with that should be a clear set of objectives of why you're doing this, what you're actually trying to achieve, and accompanying this with monitoring throughout. So again, because of that biennial life cycle, it's really tricky to see how it's actually responding. So setting up almost from a research perspective, essentially monitoring plots that you're going back to, you're doing actual sort of counts of different garlic mustard life stages and sizes and whatever factors you're interested in year to year, so that you have actual quantitative data to back up whether the interventions that you're using are actually achieving something. And finally, consider also what happens after control. So this is kind of a general recommendation across weed management in general is often there's so much focus on that removal of the weed, there's not enough thought given to what happens next. And really what we should be looking at more often is something like more of a restoration approach. There's a concept that I like quite a bit, which is this idea of this weed-shaped hole that we need to look out for. So if you are successful in removing a weed, there is now some ecological niche space there that other plants can potentially take advantage of, and they may not be the ones that they want. So it could be other weeds, it could be the same weed being able to reassert itself. So what we really want to do is focus on plugging that hole with something else. So in the case of garlic mustard, this BMP talks about, for example, mulching approaches to try and prevent additional seedling growth or dense overplanting with other native understory plants that have some ability to outcompete garlic mustard if they are planted densely enough. And there's some nice lists of options available for Ontario, at least, probably for other locations as well elsewhere. So again, this was kind of not the piece of the puzzle that we focus on ourselves so much, but I did want to give a brief overview of it. If you're interested in looking for more about this side of things, especially the management piece, please check out some of these very, very useful resources and others that are available. The second chunk that I want to look at is some work that we've been doing over the last few years, kind of an emerging research example, looking at insects and garlic mustard. The broad question that I want to sort of focus on for a little bit here is this idea of how much do we still not actually know about this ubiquitous weed that's been present for hundreds of years, that we've been managing for a good amount of time. We talk about garlic mustard a lot, so we'd sort of assume that we know kind of everything that there is to know about it. But that's very much not the case, of course. In fall 2020, I was working in Eastern Ontario. We were looking for weevil larvae in garlic mustard stems. We were collecting stems and cutting them open. This is a part of the biocontrol program that I'm going to describe later, so we will get back to this. We were cutting these stems open. We didn't find the weevil larvae in that particular instance, but instead we found something else. We found this really light granular frass and these unknown larvae that we initially had no idea what they actually were. So we did a little bit more searching after this initial discovery. We collected approximately 363 second-year garlic mustard stems, dried them out, washed them, and then cut them open to see what was inside. This was from 2 sites in that Eastern Ontario kind of region that we were working in. We found that about 14 to 27% of those stems had damage in them, so kind of that tunneling through the roots and stem structure and some frass in it. And then of those damaged stems, a good chunk, about 27 to 40%, had these unknown larvae in them, all the same type, and then even more than that had that frass present. So we were definitely looking at something that we had just kind of discovered here, weren't aware of previously, and wanted to look into further. We sent samples away for genetic identification, DNA barcoding to colleagues, and this identified this as Mortallina ancilla. This is a species that is known to North America already. It's a widespread, fairly widespread tumbling flower beetle. Mordelids in general are mono, oligo, or polyphagous, so they will eat either, they'll be very specific to one species or may feed on a variety of different species. They tend to bore through stems. They may eat other insects as well. In general, the literature on Mortallina ancilla is extremely limited. There's some very confusing taxonomy about how it's been named over the years and very limited description of its life history and ecology. Most notably, this was, as far as we've been able to tell, the very first record on garlic mustard, even though we'd been looking at garlic mustard for basically hundreds of years at this point. So following these initial findings, we wanted to look into this further. It wasn't our main focus, so this fell to Thomas Hall, who was an undergraduate thesis student who I was supervising at the time in summer and fall of 2021. And for his thesis, Thomas was looking at these primary questions of how common is this Mortallina ancilla in garlic mustard within and between sites here in Southern Ontario, and then just fill out some basic details about the life history of it and other diagnostic indications of what we're looking for if it is present. How do we know that it's there? This work consisted of a geographic survey of 8 sites across Southern Ontario to determine this overall kind of distribution and see how common they were. So essentially looking at one point in time across 8 different sites, collecting a bunch of these stems, cutting them open to see what was inside. He also did a longitudinal survey at one site through time. So going back to the same site every few weeks up to a month, sampling again and again so that we can fill out over the course of the year when they are present in different life stages. It's a little bit difficult to make out here just with the color scheme, but these are those 8 sites with pie charts representing larval presence and absence. So from this geographic survey, the basic takeaway is that Mortallina ancilla was found in garlic mustard at all of the sites that we have ever looked at so far, and that now goes beyond these 8 initial sites as well. Average stem occupancy was about 21%, but that ranged quite a bit from 2% all the way up to 72%. So again, main takeaway was that it was at all of the sites and was relatively common. So when we cracked open stems, on average, 1 in 5 had this previously sort of undescribed, undocumented larva in it. This is now a slightly older figure. We have now more data to kind of fill this out. That middle orangey-ish tan bar on the stems is showing Mortallina ancilla larvae. Basically, across the different life stages of eggs, larvae, pupae, and adults, we've now found these mordelids basically year-round in these second-year garlic mustard stems. They're mostly in hollowed out dead stems, so they seem to be using that primarily for habitat, though they also feed on the old dry sort of plant tissues inside that stem as well. From our perspective, we're happy to see that there was minimal suspected overlap with the activity of our primarily root crown feeding biocontrol agents. That's kind of what we were most worried about. So they're sort of in a different part of the stem at a different time doing their own thing. Thomas was able to learn quite a bit of other information about this as well. Again, this hadn't really been described before, so lots of great exploratory work that could be done. You see in the top left there, we found, for example, they're more likely to be found in larger stems, so that top bar in that plot there is where larvae are found. Those tend to be stems that are wider than stems where we don't find them. We can also see that they tend to be associated with various kind of stem features and appearances. So we were able to identify that those little brown granules, the frass, the insect poop was coming from them. So when we find them, we find the poop and vice versa. So we sort of learned what to look for. Importantly, we had sort of focused on mordelids, and this particular species was the most common in about 21% of the stems. But importantly, there was a whole slew of other previously undescribed or otherwise poorly described insects documented in garlic mustard as well. So lower percentages, but present. We had braconid wasps, there were various flies, thrips, other beetles that were present, and again, not really documented in garlic mustard stems up to this point. So this was, for us, a really interesting case where this biocontrol research kind of unintentionally discovered an unknown association between these relatively common species. And so I think there are a number of lessons that we can take from that in general. Broadly, you really don't know until you look. The idea that this absence of evidence of something is definitely not the same thing as evidence of absence. And from a research or natural history perspective, that there's just a lot of value in kind of keeping your eyes open and documenting things where you can, because it might be new and it might be important. And I would argue that these kind of unknown insect associations are really important for weeds like biocontrol that we're looking at. They're a piece of the puzzle in terms of understanding garlic mustard population dynamics, why they succeed where they do, why they don't succeed where they don't. There's the potential for them to be helpful in management. So they may be something that is having a negative impact on garlic mustard populations, and we want to maybe help that out. Or they may hinder management. So for example, they might be natural enemies or competitors of biocontrol agents. They weren't in this case, but that's an important thing to check for. So basically, there's a lot going on in there that we're not really aware of until we start looking into it. Okay, so the last section that I wanted to touch on then was this new management tool in the form of biological control. Biological control, or biocontrol more colloquially, is something that we like to think of as presenting another option in the toolbox. At a broad level, biocontrol refers to the use of live organisms to suppress and introduce pests. Why use biocontrol? The main idea is that we are reuniting pests with their natural enemies to manage their population. One of the big reasons that weeds or other invasive species manage to do so well in these new environments is that they've left behind some of these natural enemies that would normally keep their populations in check. So by reintroducing those natural enemies, we're reestablishing some of that ecological balance. A lot of effort now goes into ensuring that biocontrol agents are highly host specific. So they're only going after the targets that we want them to go after, which means that it's very low environmental impact. It's very specific in contrast to something like a broad spectrum herbicide, for example. When it works, biocontrol can be self-sustaining as the organisms reproduce and track the host through space and time. And because of that self-sustaining nature, it can be very cost-effective as well at large spatial and temporal scale. So you release, and you probably want to do multiple releases over different areas, but they do some sort of spreading on their own as well, which is very helpful and helps keep costs down. Biocontrol is a slow process, both in terms of impact, but also in terms of its development. So biocontrol for garlic mustard has been in development for over 20 years now. The search for suitable insects began back in 1998. There was a long period of time devoted to host range testing and permit preparation. So again, ensuring for the most part here, that time was dedicated to making sure that they're going after garlic mustard and garlic mustard only. This culminated in a Canadian permit to release that was approved by the Canadian Food Inspection Agency in 2018. And that then kicked off the first Canadian releases and the first sort of pilot scale program for this in Ontario here in North America. And our lab and colleagues with Ag Canada and others have been involved with it since that time. Biocontrol of garlic mustard in North America uses a European root crown mining weevil, Pseudorhynchus scopicollis, shown here. I think they're quite cute, but that's sort of a subjective assessment. Larval root crown mining is kind of the main form of damage. So we see on the right, there are some of the larvae sort of digging through the root crown. So right where the leaf stalks meet the ground, you have a chunk of fleshy tissue there. So they'll develop in that and feed on it. And that reduces garlic mustard survival, growth, and reproductive output. So it can either kill the plant or just reduce its overall competitive ability and allow other plants ideally to stand more of a fighting chance. You also get some very minor adult feeding on the leaves. So the weevils will sort of feed on leaf tissue. You get this very characteristic window painting that you can see in the center there, where they've gone partway, but not all the way through the leaf there. This doesn't really have a major impact on the plant, but is something that's visible as well. You can see some figures taken from Jeannie Katowicz's led NASEMA biocontrol sheet here with much better photos than I have. You can see the different life stages, including the egg in the top left. These eggs are laid into the leaf tissue of the leaves and the leaf stalks and are quite small, difficult to spot, but they're there. You can see another one of those larvae mining through that root crown, and then some examples of the adult weevils there. This is the full version of that life history table or development table that we're seeing for both garlic mustard and the control agent, Pseudorhynchus strobicollis here as well. It's kind of a complicated life cycle, so we're going to walk through this, starting with adults emerging from the soil. This happens sort of in May, June. They'll feed briefly for a period of time there. That's where that hatched bar is. And then they go dormant for a period of the summer. They don't like particularly hot, dry conditions. These adults will then sort of wake back up in the fall. They'll start feeding again, and then they'll start laying eggs in the leaves and the stem. And they'll lay eggs all the way from fall through to the following spring, through the winter, when conditions are warm enough. So they'll sort of be active and then slow down and then start up again. So you can get eggs being laid all through that time. Those eggs can then sort of start hatching when the conditions are right, shortly after they've been laid, so you can get larvae active as early as the fall, and they will make their way down and mine the root crowns, again, from that fall through spring period. So they go through the winter either as eggs or as larvae. The larvae will then exit the plants in the spring and pupate in the soil. They make a little casing out of soil materials for themselves and then emerge from the soil in the spring to become adults at that time. And then that takes us back to the beginning of the cycle as well. So it's a tricky system to work with in terms of garlic mustard has a fairly tricky life cycle, and then the weevil has a tricky life cycle as well. But here we can see it all laid out. The primary research goal for this has been to implement the first biocontrol program for garlic mustard using Pseudorhynchus strobicollis. Because this is the first time this has been done, our focus right now has been on really development of a lot of core background methods. So we've been doing some background monitoring of garlic mustard populations to understand its impacts and demographics, how the populations grow and recede. A lot of our time has and is still being devoted to developing these core methods for how we rear the insects, how we produce them, how we release them onto the landscape, and then how we monitor, hopefully, their population growth, spread, and impact. And by releasing, what we're also trying to set up are what we call these nurse sites. So these are areas where populations of the biocontrol agents are doing well, they're growing, and these are populations that we can then go back to and draw insects from for further redistribution to other sites or to support further rearing. So we're going to step through, this is kind of a bit of a picture-based show and tell primarily of those 3 different steps in the process to give you an idea of what this actually looks like. And I should say that these are all methods that we're still very much in the process of figuring out. It's an iterative process of trying different things, seeing what works and redeveloping it. And I'd say we're getting close with it. So first you need to produce more insects. So this rearing process begins with the shipment of weevils from collaborators with CABI, based in Europe, in Berlin specifically, and they are sent to us, and then we have to rear them through 1 generation prior to release in Canada. This ensures that we are putting out weevils that have been reared under quarantine lab conditions, so we're not accidentally introducing any other parasitoids or pathogens with them. So they're 1 generation separate from the field European weevils. So you can see some of the weevils in a styrofoam box here, kind of like how we would sort of ship them. They're usually a bit better packaged than this, and they have some garlic mustard leaves there to feed on and keep them going. We then mark the parental weevils with a UV fluorescence, so sort of a paint pen or just a modeling paint. Just a little smear on the elytra. So these are paints that will glow brightly under UV light, which makes them much easier to spot when we're trying to collect them off of plants. Otherwise, they're very good at hiding. So it's a lot of work to mark them, but it's even more work to pull them off plants without this marking. So you can see on the right there, that's under ultraviolet light. one of these weevils sitting on the other side of a leaf with some of that fluorescence marking very visible there. We then need to expose garlic mustard plants that we have either collected from the field or reared, germinated from seed ourselves, to sets of mating weevils. We've done this a number of different ways over the years. On the left, you can see plants in individual containers, so we need to contain them so the weevils don't just walk away. Or in sort of larger pots with mesh bags set over them, which you can see on the right, which is what we're sort of favoring now for the most part. After about a week, we retrieve the parental weevils and then use them to expose other plants as well. So that's why we need that ultraviolet marking so that we can go into these plants, spot these tiny little brown dark weevils that are hiding in the soil potentially, and then move them to another plant. So again, you can see one showing up against the dark soil there with that UV fluorescence down near the base of the plant on the right. If this seems like a lot of work, it is a lot of work, but we're getting better at it, I would say. Once you have exposed plants, so from that life cycle table, we can see that they have those eggs or potentially larvae in them if they've started to hatch. We can either take those plants and release the plants themselves, and we'll look at release in a second, or we can overwinter them and then collect the emerging offspring larvae, or that should say beetles, actually. Typically, we're looking at beetles as they emerge from the soil and then can be trapped using methods like this, where we're putting the whole sort of plant in a box so it's dark, and then giving them some fresh garlic mustard leaves in a cup that they will be attracted out to, and then we can collect them for rearing or release or whatever we're going to do. Once we have the weevils, we obviously need to release them onto the landscape. Now that we have these parasitoid-free, containment-reared weevils, we release them at field locations to try and control garlic mustard, but also at this early stage, really what we're doing is trying to establish these nurse sites, just try and get a population going and figure out those core methods. There are two main methods we've been using for release so far. The first, as I mentioned, is to transplant those plants that have eggs and larvae in them directly into the field in the spring. You can see some plots where that's been done, where those pots have essentially just been trenched into the ground, and they'll start to have some regrowth, and the weevils will emerge from them over time and then switch to plants in the field. This has worked, but does involve a lot of disturbance of the release sites because of all this digging and soil movement and so on. The second release method requires more rearing effort because we have to get all the way from weevils to a second generation of weevils, but it's much easier after that, which is basically just dump the weevils out into the field. So maybe we transfer some leaves that they've been feeding on from a box. You can see that in the left there, and eventually they'll wander off and just switch to plants into the field. So you can see one of them highlighted on the right there. And this can happen in the spring or in the fall. Basically, we're just trying to avoid that summer hot, dry period that they really don't like. And finally, once they're on a site, monitoring becomes the main activity. You're revisiting these release sites to assess the biocontrol agent's ability to establish, and then over time, eventually to spread and ultimately have an impact, hopefully, on the garlic mustard populations as well. So generally, this involves searching for different types of insect activity around the release points. So you can see on the left there are some corners of plots where we've released weevils, so we can intensively monitor within those plots. But we also want to track how they move away from those plots as well. So on the right, you can see we're going from the corner of one of those plots, running a transect up a hill into the woods further away, and we can monitor how far they've moved over time. Generally, what we're looking for, the easiest thing to spot is that characteristic window pane feeding damage that we get from the adults on the rosettes. Once you sort of train yourself to look for it, it can be fairly easy to spot, but it is a very indirect form of evidence. Ideally, for direct confirmation, we want to go in and get some weevil larvae or adults themselves. So we can do sweep netting or aspirator collection, or go in and collect and dissect roots. That's what we were doing when we found the mordelid larvae earlier. This all requires a lot of work and more disturbance of the site. So we kind of minimize this early on when we're trying to get populations to establish, but ultimately we want to get some confirmation somewhere in the process as well. So using these methods, which we're still in the process of developing, since 2018, we've released a total of 62 weevils and 115 infested plants. across 4 sites in Ontario. If you're familiar with other biocontrol projects, including our work on Phragmites, you'll note these numbers are a little bit low in comparison. And that just reflects the fact that this system, these biocontrol agents, take that much more work, time, and effort to really sort of rear and put out there. But these numbers are growing over time as well. I'm not going to show any data from this, but we do have some promising, very promising initial monitoring evidence. We're seeing adult feeding at quite a few of these sites, and we've also found some larvae and adults that we're just confirming are indeed our species of interest. And these are all good signs indicating persistence at the field site once we put them out, that they're surviving, and also that they are reproducing. So once they get through at least one full life cycle and produce offspring, that's a really good sign that they're able to do that in the field here. So this is really early days of this, but again, we're moving forwards and we're going to see more activity on this in the years to come. We're going to keep monitoring all of these past releases, and we'll be focusing on new rearing and releases going into 2023. Currently, we have approximately 340 infested plants with eggs and larvae that are being overwintered here and in Alberta as well, that we are going to be releasing this coming spring and summer. So I want to very quickly outline 4 general reminders about biocontrol before we finish up here that apply to this system or any others as well. For those not as familiar, it's important to remember that biocontrol is very much a long-term solution for large-scale problems. It's slow to implement, it's slow to have an impact, but that all goes in hand with many of the benefits, that it is self-sustaining, that it's sustainable across these large spatial and temporal scales. So if we think of success stories like purple loosestrife biocontrol, this is something that develops over time, and a garlic mustard biocontrol program would be no different. Importantly, the objective is not complete eradication, and it's not what we're expecting, and it's not what we're hoping for either. The realistic goal we're trying to achieve here, it's added pressure against the weed. We slow spread and limit spread, decrease its competitiveness, and this can combine with other management or just lead to a situation where we have garlic mustard still on the landscape, but at a much less damaging density overall. We talk a lot about risk and biocontrol, and there's a lot of steps and extensive host range testing that is taken to mitigate risk, but it's important also to compare risks against the risks of other management or of doing nothing. So any kind of activity, whether it's burning or broad spectrum herbicides, for example, have risks that we weigh against what's going on here. And importantly, doing nothing at all involves a certain amount of risk to these habitats as well, where we're losing key biodiversity and ecological function as well. So all of these things need to be factored into that risk calculation. And finally, it's important from an expectation level to remind that biocontrol is not a panacea. It's not a silver bullet. This is an additional option in an integrated pest management toolbox. So it's something that is meant to be used alongside other tools. Having different tools gives you flexibility to deal with different situations. And that's very much what we're trying to offer with this here. Okay, so let's wrap up with a few quick conclusions. We covered a fair bit of ground there. We described garlic mustard as this, of course, widespread understory plant invader. We looked at some of the factors that makes it successful as an invasive weed, and we considered some management approaches, including other resources that are good to look at for pursuing aspects of that further if you're interested. We looked at this idea, the importance of acknowledging and trying to fill some of these ecological knowledge gaps that we can have forming around even very common weeds that we've had for a very long time. And we looked at that through this case study of this very first description of Mortellina ancilla and other insect associates showing up inside a high percentage of garlic mustard stems if we just crack them open and actually look for them. And then finally, we looked at biocontrol as a new emerging sort of tool and that we're trying to add to this toolbox to help manage invasive garlic mustard. And you're going to be hearing more about this in the years to come as more and more of those core methods are sort of locked in, and we're able to focus increasingly on increasing the scale of rearing and release on the landscape. So of course, we've talked to a number of different projects here over a number of years, and all of this wouldn't have been possible without the support from many collaborators and supporters, both institutions and individuals. So a big thank you to everyone who's been involved with that throughout. And again, thank you very much for having me here today. Happy to take questions if we have a few minutes now. And also, please feel free to get in touch with me here. That would be my work email, which would be the one to use if you have any questions.
Thank you so much for that presentation. It was great.
Cool.
We'll be sending out an email with the webinar recording and with an educational handout that will include a lot of the resources that Dr. McTavish included, so you can look forward to that. And just before we jump into the questions, I wanted to let everybody know about our survey. So Monica will add that link in the chat. So just let us know how you enjoyed the webinar today, and if there are any topics that you'd like to hear in the future, we greatly appreciate that. Okay, so I do see some questions in the chat, and if anyone thinks of any more, you can add them in now. So I'll read out the first one. So it says, what native vegetation does C. scrobicalis usually eat? Is it all herbaceous species, or does it include woody species? And does their diet include agricultural species?
Right. So Pseudorhynchus scobicollis, which is the biocontrol agent that has been permitted for use against garlic mustard in North America, is host specific to garlic mustard, Alliaria, petiolata only. It is specific to that species, and it's the only thing that it's been documented to eat. So that's known from its home range in Europe. So where it lives with garlic mustard, that's what it completes its life on. And there have been many, many years in the lead up to that permit that have been spent with choice and no choice testing to sort of say this is the host that it uses, and it doesn't switch other things. So there's not any evidence to support a risk to any other kind of vegetation, whether that's crops or naturally occurring native herbaceous species or woody species either.
Perfect. And then the next question is, I'm afraid to pull garlic mustard, but I'm concerned about what will happen if I don't. What do you suggest so I know how to properly and safely take action?
Right. Yeah. And that is definitely a dilemma. And, from that management portion of this talk too, I don't want to give people the impression that, it's a hopeless thing, or that it's even deleterious to sort of be doing something about it. I think it's more about, considering some of these factors about the importance of really being sustained in your efforts to control, and evaluating the different, even just kind of physical kind of options that exist in terms of pulling versus cutting or mowing, sort of cutting seed heads. The main thing you're trying to do, of course, is try and prevent more seeds from getting into the population. So cutting seed heads at a certain point in the development, so that you're not getting as much disturbance to the soil, but you're preventing that input into the seed bank that's ultimately needed to maintain that population is another option to sort of pursue. So I would, again, we don't work on those management efforts ourselves, so I don't want to speak too much to something we don't have familiarity with firsthand. I would direct you to, again, some of these best management practice guides to look at the different kind of sort of individual landowner scale management options that are available, like cutting and mowing and pulling, and sort of evaluate what seems to be most suitable for your sort of situation. What I would generally sort of say, again, as the general advice is keep it up for a number of years, and if you have any capacity to actually sort of count or track how the population actually responds to this over time, that can be a way to gauge, is this actually having a positive effect, or is it maybe making things worse and should be discontinued? So maintain it, sort of look at the options that are out there, and sort of collect even just very basic data about how this actually changes over time. And good luck.
And the next question, if a defined area has been cleared of garlic mustard, over what distance might seeds travel to repopulate that area within a seasonal cycle? In other words, how big of a margin should you aim for between a supposedly cleared area and nearby areas of infestation?
Right. Yeah, that's a really good question. I don't have a great quantitative answer for it. I know there are sort of figures that exist. Again, I think that OIPC BMP may have some sort of a spread rate on that. I know for second year plants, so where the seed actually comes from, the seed rain falls pretty locally. So, if you think of a dried out garlic mustard plant with its saliques, they don't take to the wind. They basically just kind of drop off. So they tend to land, within a few meters of the plant. Wind can contribute to that, of course, as can overland water flow, or animals, or sort of material that gets caught up in boot treads of people walking through an area can certainly contribute to longer range dispersal. But barring all of that, it tends to be a fairly sort of local effect. So as long as sort of things are being you're being cautious about moving through the area and not moving material back and forth between them. Sort of the buffer area doesn't necessarily need to be massive to achieve that kind of isolation that you're looking for. Again, I would look to the BMP. I don't want to throw out a specific number, but something on the order of, a number of meters or 10 meters, something along those lines would probably be sufficient based on our understanding of how far these seeds travel on their own.
Perfect. And just one last question here. What effect does garlic mustard have on pollinator populations?
Right. So the main effect is sort of an indirect one, which is primarily that, by replacing other herbaceous diversity, you're sort of depleting the other diversity that's available to support those pollinator communities. So if you're taking a diverse understory population and replacing it with something that is almost exclusively garlic mustard, and that garlic mustard is not a suitable for those pollinators, then the pollinators lose out in that regard. There's also some data about how garlic mustard can be a pollination sink for some species that sort of are attracted to it, and an oviposition sink where they will sort of lay eggs there, but it's not able to support that life cycle. That's a little bit more species specific. For the most part, I would say the impacts stem from the fact that it is outcompeting those other plants that are needed to support those pollinator communities. So we have a net, just blanket negative effect on those pollinators.
Perfect. So that concludes our questions. So don't forget, everyone, to register for our upcoming webinars this week about other invasive species and fill out our survey. Thank you, everyone, for joining us, and we hope you have a great rest of your day.
Thanks, everyone. Take care.